Okay, today we're going to start out with a scholarly lecture on protein synthesis, and we're going to look at all aspects of it. It all starts with DNA, so let's take a look at this short video.
Vote for nucleic acid. We've been here since the beginning of time. We are what make you who you are. We make up the DNA from your father's father's father to your father's father, or your mother's mother's mother to your mother's mother, or even your mother's father's mother's mother's father. We make miracles happen. And what are lipids? Anyway, so much of them can make you die. And carbohydrates? Ever heard too much carbs is bad for you? Don't get me started on proteins. They might seem like they're important, but man, no, we just take it in nature like there's no tomorrow. They won't be there when it's a little too hot. Pansies, but we will. We are the future. We are your neighborhood-friendly, loving macromolecule made of nucleotides bonded by phosphodiester linkage. If you trust yourself, you can trust us. We are the recipe to your existence. Vote for me if you want to live. This message has not been approved by actual nucleic acid.
That's a cute little video. The reality is we will be talking about protein synthesis, but we have to start with DNA. The process requires several steps, and there are several concepts within the process that we want to review, especially those that go back to chapter 2 and on the cellular organelles. Remember that? The DNA is in the nucleus. The nucleolus produces RNA, both messenger RNA and ribosomal RNA. Oh, and the third one, transfer RNA. All of those are involved in the process. So the process is controlled by DNA, and proteins are manufactured by RNA, 3 types. So nucleic acids are very important in protein synthesis. The first part of protein synthesis is code. It's transcribing the code. We're all familiar with Morse code, right? So if I go ... dash dash ... most of you know that means SOS. Code is language, and language is taken for granted by most of us most of the time. Do you realize that mathematics is a language? I hope you do. What we're going to find out is that DNA has a language, and it has to be transcribed into code before we can direct the making of proteins out in the cytoplasm. In order to start the process, DNA has to send a message in code out to the cytoplasm. To do that, it has to transcribe that message into code. The animation on your PowerPoint that we'll look at quickly here demonstrates how this is done.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches a specific sequence of nucleotides on the DNA called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.
So as you can see from this first animation, the first step in protein synthesis is to send the code with an mRNA molecule through the wall of the nucleus and into the cytoplasm, where it will hook up to a ribosome, and a ribosomal RNA was made in the nucleolus also. And when it hooks up with the ribosome, that will form a sort of a biochemical factory that will begin to make the protein using enzymes and transfer RNA that was also made in the nucleolus and that is now very specifically floating around in the cytoplasm and has attached to specific amino acids that will be brought in the process of making the protein. And that process is called translation.
After the turn of the century, the clear blue skies over Germany came a roar and a thunder that I never heard, like the screaming sound of a big.
So at the beginning of that version of the Snoopy song, you heard Achtung, Achtung, jetzt singen wir alle zusammen die Geschichte über die Schweinhund, wir halten für den Namen Red Baron. Do you know what that means? In order to find out what it means, you have to translate it. So it's a cute little song, isn't it? It means, attention, now we're all going to sing together the song about the person we call the Red Baron, or the Schweinhund that we call him, the Red Baron. And that's what's going to happen now out in the cytoplasm. Messenger RNA floated out, as I mentioned before. It attaches to ribosomes. In fact, many molecules of messenger RNA will probably go out there and activate a number of ribosomes simultaneously so that we can produce lots of molecules of protein. We're not going to do just one molecule. We're probably going to produce a few million of them, actually. So let's go out into the cytoplasm next. And let's take a look at an animation from the PowerPoint that demonstrates what translation is and how the proteins are actually formed.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, A new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site, as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
This table, which is in the PowerPoint, is also on page 201 of your textbook, and it shows the different codons coming off of the tRNA that code for specific amino acids. There's 64 different codons, but there are 20 amino acids. Count them. 1, 2, 3, 4, 5, 6. 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Some are replicated. It also shows the redundancy, so that when mistakes are made, sometimes the mistakes aren't so bad because different codons, for example, here's proline, can be coded for by different sequences. So let's suppose there's a mutation. And let's suppose in this sequence, this C is knocked out and replaced with a uracil. So if cytosine is knocked out and replaced with a uracil, we still get a proline. So that gives your body some protection against random mutations. And that's sort of an interesting thing. Finally, an important aspect that we have to talk about is that once this protein is made, it can't function just lying flat like a piece of string. It will biochemically fold into a structural unit, something like a jigsaw puzzle that will fit somewhere else where it can conduct its functions. But it is structure that is very important in all of enzyme proteins. Structure is function. That's why when we denature proteins, they stop working. When we get them too hot or cook them, they can't do their job anymore. Let's take a quick look at a video. It's less than a minute long by Gabriel and see what we're talking about. Proteins are going to fold to become functional.
Proteins are polymers constructed from the same set of 20 different amino acids. The primary structure is the specific sequence of amino acids specified by the gene. In the secondary structure, portions of the protein begin to coil and fold into unique three-dimensional formations. Two kinds of secondary structure units are the alpha helix and the beta-pleated sheet. The folds and coils of the protein are the result of hydrogen bonds that occur at regular intervals. The tertiary structure is formed by interactions between side chains of various amino acids. At this stage, some proteins are complete. Other proteins incorporate multiple polypeptide subunits, creating what is known as the quaternary structure.
So as you contemplate what we've been talking about, protein synthesis, one of the exercises I'd like you to be able to do is to take a code, a piece of DNA with several sequences of codons, so that there's 5 or 6 different amino acids involved. Take that code of A, C, T, and G, transcribe it into mRNA, replacing T with uracil. Take it, figuratively speaking, to the ribosome, and then transcribe. Transcribe is what you did from the DNA when you made the mRNA, and then on the ribosome translate it into the protein by stringing the amino acids together